DNA replication is a semi-conservative process in which each strand of the DNA double helix acts as a template for a new complementary strand. But what specifically happens for this to occur? First, let's look at the DNA nucleotides and what makes them. DNA nucleotides are made up of 3 components: a phosphate group, deoxyribose, and a nitrogenous base. There are 5 different nitrogenous bases that can be separated into 2 categories: pyrimidine bases and purine bases. Thymine, cytosine, and uracil are pyrimidine bases. However, uracil is a base in RNA, not DNA. Adenine and guanine are purine bases. How do they bind together? Thymine bonds with adenine with 2 hydrogen bonds, and guanine binds with cytosine with 3 hydrogen bonds. Now let's take a look at replication. Here we have a DNA molecule with 12 sets of nucleotides. Before new strands are built, helicase separates the parental strands, opening DNA up. After this, primase binds to the template strand and synthesizes a small strand of RNA called a primer, which attaches to the 3' end of the parent strand. The primer is used to begin the new DNA strand. Then DNA polymerase synthesizes the new DNA strand for the leading strand first, which is the strand that runs 5' to 3' towards the fork. Then to the lagging strand, which runs 3' to 5'. Now the new strand is made but not yet attached. During this time, Okazaki fragments are also formed, which are fragments of DNA being synthesized on a lagging strand that aren't fully attached to each other. Ligase, an enzyme that joins adjacent fragments, seals off the DNA, once again starting with the leading strand and then the lagging strand. The ligase also seals off any Okazaki fragments that are left on the lagging strand. Throughout this whole process, DNA can easily get wound up, so the enzyme topoisomerase relieves the stress from unwinding, ensuring that finally replication is completed, and there are now 2 new strands made from the parent strand. In prokaryotes and eukaryotes, DNA is different. In eukaryotes, DNA is a single circular chromosome, and replication occurs in the nucleus. In prokaryotes, it exists in multiple linear chromosomes, and replication occurs in the cytoplasm.
After DNA replicates, the next step is transcription. Transcription is the process by which DNA is converted into messenger RNA. Transcription has 3 main parts to it: initiation, elongation, and termination. Transcription begins with a double-stranded DNA molecule. A key player in this process is an enzyme called RNA polymerase. RNA polymerase binds to a specific region of the DNA known as the promoter. This interaction opens a small section of the DNA, exposing individual nucleotides and initiating transcription. Once transcription is initiated, RNA polymerase begins to synthesize new RNA nucleotides using the template strand of the DNA. It reads the template strand from the 5' direction and adds nucleotides in the 5' to 3' direction, leading to the extension of the going mRNA strand. RNA polymerase will continue to synthesize new mRNA strands until RNA polymerase encounters a specific sequence on the DNA called the termination site. Upon reaching this site, transcription stops, and the newly synthesized mRNA strand is released. RNA polymerase then detaches from the DNA, allowing the double helix to reform with itself. In comparing DNA to RNA, they share many similarities with DNA, but there are crucial differences. mRNA is single-stranded rather than being double-stranded like DNA. RNA is composed of repeating nucleotides that includes a 5-carbon sugar molecule called ribose instead of the normal deoxyribose on DNA. RNA does include phosphate groups, and notably, while mRNA includes the normal nitrogenous bases guanine, cytosine, and adenine, it replaces thymine with uracil. Once transcription is complete and mRNA has been synthesized, it exits the nucleus and enters the cytoplasm where it will undergo translation. In eukaryotes, transcription occurs in the nucleus in the cell before it's transported into the cytoplasm for protein synthesis. In prokaryotes, transcription occurs in the cytoplasm of the cell since prokaryotes don't have a nucleus.
The final step is RNA translation. RNA translation consists of many moving parts, including ribosomal subunits made up of rRNA, amino acid carrying tRNA, nucleotide carrying mRNA, codons, a polypeptide chain, initiation factors, and termination factors. The goal of RNA translation is to direct the addition of amino acids during protein synthesis. The first step in RNA translation is initiation. To start the process of RNA translation, a small ribosomal unit binds to mRNA. tRNA then carries the amino acid MET to bind to the start codon of the mRNA sequence, which is AUG. The 5' start codon of AUG must be recognized for translation to begin, and the position of this start codon will determine how other codons are recognized downstream along the mRNA sequence. This is caused by the attachment of the protein initiation factors that bind to the mRNA and small ribosomal unit. When the initiation complex moves along the mRNA and reaches the AUG start codon, the large ribosomal subunit joins the complex. Once the large ribosomal subunit has joined the complex, the protein initiation factors are released. RNA translation differs between eukaryotes and prokaryotes. In eukaryotes, the initiation complex will form at the GTP cap and scan along the tRNA strand until it reaches the first AUG start codon. Due to this process, each mRNA codes for a single polypeptide. Prokaryotes lack a GTP cap, so the initiation complex will attach at an internal mRNA sequence and then move downstream to find the first AUG start codon. This organization is called operon and allows for the coding of multiple polypeptides. The second step in RNA translation is elongation. The process of elongation is amino acids being added to the growing polypeptide chain. The tRNA with the MET start codon is bound to the ribosomal unit, and the next tRNA prepares to bind adjacent to the start codon. The amino acids produced during elongation are bound together through peptide bonds that come from dehydration synthesis. The large ribosomal subunit of RNA catalyzes the reaction, which causes a short polypeptide bond to form. The ribosome will shift one codon to the right, releasing the first tRNA and allowing the next tRNA to come into line. The process is repeated with each tRNA bringing an anticodon that reads the codon in the nRNA sequence. The codons are read in groups of 3. The amino acid on the added tRNA binds to the polypeptide chain, growing it in length. This process is repeated, and energy for these bonds come from elongation factors that gain their energy from breaking off from GTP. The final step in RNA translation is termination. RNA translation ends when RNA polymerase encounters the terminator. These terminators are stop codons that the tRNA molecules cannot recognize. At this point, the release factor binds to the ribosome. This causes the polypeptide chain to release and the small and large ribosomal units to dissociate from one another. All components of RNA are separated. There are three kinds of DNA mutations. The first is insertion, where an extra set of nucleotides is inserted. The second is deletion, where a pair of nucleotides is deleted. And the third is base substitution, where one pair of nucleotides is substituted for another. All three of these result in changes in the sequence, which then affect the DNA.